Okay, a few little bits and pieces about nuclear structure. Now, there appears to be some evidence that their nucleus may have some type of internal substructure. Some kind of internal organization consisting of proteins, possibly and/or RNAs. It does not appear to be something like a nuclear equivalent of a cytoskeleton, although it's interesting enough that it's been seen enough times in enough cells that there is evidence that there's actin, the protein that makes up microfilaments, that there's a significant amount of it in the nucleus. So it may play some kind of role in structural organization there. But the nucleus and nuclear structure has a number of things. Whatever it is, it's kind of maybe not as well defined as what we see in the cytoplasm, but there does seem to be something there. I want to point out a couple aspects of things like that. And the big thing is, what's doing it? That's where you have the big debate. Okay, for instance, if you take a look at the nucleus, you could use immunofluorescence, labeled antibodies, or you could use hybridization, what we call in situ or in-place hybridization for nucleic acid sequences. And you could identify certain things where certain things might go on. For instance, let's suppose we made antibodies against small nuclear RNPs. In fact, there's plenty of them around. In other words, you want to find where are the spliceosomes? Or you made antibodies against key parts of the basal complex and ask, where do we see that? Now, what you might expect if you did an experiment like that, you might expect that you would find labeled stuff throughout the entire nucleus. In other words, that splicing is occurring everywhere in the nucleus. All you need is something like, messenger RNA precursors or things like that. You might figure transcription's occurring anywhere there's genes that are supposed to be transcribing stuff. What you find instead is something different. And it's hinting that there's some kind of structural organization in the nucleus. You don't find splicing occurring all over the place. Instead, you find these little small fluorescent speckles in discrete locations in the nucleus. And if you're looking for things involved in transcription, you find them in these kind of little speckles. And if you look at splicing, you find them in a different set of speckles. And occasionally you even find some overlap, where you might find certain speckles have components for both transcription and for RNA processing. So, it's telling you there's a kind of distribution. Instead of these, it appears that instead of transcription, and instead of splicing being randomly scattered throughout the nucleus, it appears there's discrete splicing centers. And there's discrete areas of transcription, and maybe what you have to do is effectively move the chromatin to one of these splicing centers or one of these transcription centers, instead of just occurring wherever the appropriate genes are. You have to move these genes into the splicing center or into the transcription center. So there's evidence for that we have distinct areas in the nucleus that are full of the appropriate things to do transcription, splicing, whatever the case is. That's kind of neat. Sounds like it's hinting that there's some kind of structural organization in the nucleus. Now, a little hint, because we're going to go into this later. It is possible that a major factor in this are a class of relatively recently discovered RNAs called long non-coding or LNC RNAs. But we'll touch on those guys later. They may play roles in assembling all this stuff together in discrete locations. So, we have possible or probable, a little aside on this, a lot of this work was pioneered and still being pioneered by this person at Cold Spring Harbor named David Spector, who happened to be on the outside person on my thesis committee. So he was pioneering this work of using labeled antibodies against nuclear processes and finding that they occurred in discrete little speckles, discrete regions in the nucleus. Yes?
What's that top word?
Top word.
P.
This?
No.
Oh, probable transcription centers. I flunked it in elementary school consistently. It hasn't changed. And splicing
At least
centers.
you're consistent.
Oh, yes, yes, absolutely, yeah. Hey, you can't teach an old dog new tricks like legible handwriting, right?
You can.
Not me. They threw hands up in despair in 6th grade and declared me officially a lost cause of
It
matter.
just depends on the dog.
Well, not this dog. There's all things you might hear, you know. All right. Okay, so that's one kind of thing about nuclear structure. Another thing. Now this is kind of controversial, but there are some that say there is some kind of poorly defined structure called a nuclear matrix. Some kind of, and I'm going to put this in question marks because it's very controversial. And this appears that may be made of certain types of proteins, both ones that seem to be fairly common from cell type to cell type, and certain types of unique proteins, and possibly certain types of RNAs as well. Now this is not quite a nuclear cytoskeleton or anything, but there may be some kind of substructure. And interestingly enough, there are certain parts of chromosomes certain sequences of DNA that are parts of chromosomes seem to associate with this controversial matrix structure. Now what it is, and indeed is it actually real, is a huge matter of debate. People have found when you take nuclei and you strip the membranes away with detergents, detergents dissolve cell membranes, right? Okay, you do that, you get something that still looks like a nucleus. It still keeps its shape itself. Now, add in salts that strip proteins off of the DNA, strip the chromatin proteins away. Now you see all this DNA in there. Add DNA digesting enzymes to get rid of the DNA. So now you've gotten rid of all the chromosomes, all the chromosome proteins, you destroyed all that stuff. And lo and behold, you still see something that under a microscope looks like a nucleus. And that remaining stuff seems to be extremely resistant to break. And it takes extremely high salt levels, nasty protein digesting enzymes, or what have you. And that was kind of like something leading towards what is this, a nuclear matrix? On the other hand, the criticism is that a lot of times if you throw huge amounts of salt in solutions of proteins and things like that, you can get artificial aggregation of proteins. So some people say, oh, that's just, yeah, you threw so much salt in everything's sticking together, so here's your matrix, just loose aggregations of proteins. So there's debate about that, but the truth is probably somewhere in between. But anyway, the potential that there is some kind of nuclear substructure that helps to organize things in the nucleus is indeed there. The speckles and stuff, that's happening in nuclei that are not being treated in nasty ways. It's happening under physiological conditions and even in living cells. So that is real. As to what holds it in place, that's one more biological mystery. Now, one last thing I like to mention about nuclear structure, aside from all this hazy, controversial type stuff, is we've got the nuclear membrane, we've got these, oh, I hate this job. I can't use some of these. I really like to use words to describe this talk at hand, except for especially goes up on YouTube. Okay, right. All right, here's our nuclear membranes. Here's the pore. I'm not going to use nuclear pore. I'll just call it NPC. All right. Now, underneath the two nuclear membranes is a network, a thin fishnet of protein filaments. And we call this the nuclear lamina. Okay, it's just beneath the inner nuclear membrane. And those basket structures in the nuclear pores associate and bind to this underlying nuclear lamina, and it also appears to bind to proteins in the inner nuclear membrane. Now, if you look at this, it appears to be a kind of like thin meshwork of protein fibers. So here's what it looked like if you're looking top down. This is sort of a cross-section thing. The nuclear lamina is thin, but it's associated with the nuclear membrane just beneath it. The nuclear membrane has proteins that specifically bind to it. Now, what is it? Well, it's relatively easy to isolate and purify this stuff. So we isolate and purify it, and we find it's made of three major proteins that are called the lamins. So we have lamins A, B, and C. And they collectively assemble to form these fibers of the nuclear lamina. So they've identified these proteins. When they got the DNA sequence of these proteins, and they looked at it, looked at it in the databases, these fall into the category of cytoskeletal proteins called intermediate filament proteins. There are these parts of the cytoskeleton called the intermediate filaments because they're midway in size between the small microfilaments and the much larger microtubules. There's many different kinds of intermediate filament proteins that tend to be specific to certain types of cells. And they all have the property of assembling together in a certain way to make these intermediate filaments. The best example we know of are the proteins called the keratins. These are intermediate filaments of the epithelial cells. And keratins make, in fact, the outer layers of your skin are dead cells with nothing but keratin filaments left. Your hair, your nails, your claws, the horns I have when I give exams out, all those kinds of things. Well, some of you folks probably realize that, yeah. But all those things, those keratins, people call it keratin, but it's actually keratins. There's quite a few of them. Those are intermediate filaments. So now the nuclei of cells also have their own intermediate filaments. We know these things are intermediate filament proteins because all of them have certain key structural features that are common to all intermediate filament proteins. Now, intermediate filament proteins, you put them together, they instantly assemble to make filaments. How do you break those filaments down? Phosphorylate the proteins. They're regulated by phosphorylation. You phosphorylate the proteins, and what happens is the filaments break up into the individual proteins. Remove the phosphates, and they reassemble on their own. So they're regulated by phosphorylation. Okay. Well, this nuclear lamina must be fairly important in the nucleus. I'll give you an example. What do you think would happen if all of a sudden you phosphorylated those lamins, the proteins of the nuclear lamina? Well, if you phosphorylate the lamins, the nuclear lamina would quickly disintegrate into the individual proteins. And since they're connected to the nuclear membrane, you might expect that the entire nuclear membrane would collapse into little small fragments, meaning there's no boundary between the nucleus and cytoplasm anymore. Sound like a good idea? Well, every time a cell divides, that is exactly what happens, folks. One of the hallmarks of prophase of mitosis is the nuclear membrane disappears. Remember that from high school or intro bio? It doesn't really disappear. It breaks up into little tiny fragments. The reason why it disappears or breaks up is that the cyclin-dependent kinase that triggers off mitosis phosphorylates, and we know it's a specific target, phosphorylates these lamins. The nuclear lamina disintegrates and the membrane that's supporting collapses. It's kind of like those controlled demolitions of buildings. What do? They use explosive charges to cut key structural elements, like the major girders and stuff and beams, and support columns, and then those collapse and the rest of the building collapses on it, and that way you can drop a 30-story building into its foundation and not drop it all over the surrounding street. They actually did that in Cleveland, dropped a 30-story building in the middle of downtown Cleveland and didn't even break the windows in the buildings across the street. Pretty impressive work. Okay, but anyway, it's the same kind of thing. We phosphorylate the laminas. That's what causes the nuclear membrane to disappear during mitosis. Okay, so clearly, this plays an important role in organization. We also see that certain parts of chromosomes seem to be specifically attached to the nuclear lamina. Not every part, but certain specific sequences like to stick to the nuclear lamina. So it helps to organize the chromatin. But this also plays another role, because there is this fortunately very rare disease, actually it's several diseases, but one in particular is the worst, that at any given time affects probably not too many more people in all the world than you could find in a classroom or two of this size. And this disease is called Progeria. You may have even heard of it. The hallmarks of progeria are what looks in many respects like vastly accelerated premature aging. The few children born with this start showing signs that are very similar to aging in a lot of respects before they even enter elementary school. By the time if they indeed live long enough to leave elementary school, they often have look like they're little 80-year-old men and have a lot of physiological symptoms of extreme aging. Muscle weakness, organs not working all that well, weak bones, weak cardiovascular system. A lot of times they die of cardiovascular disease, in like 3rd or 4th or 5th grade. It's a nasty disease, fortunately very, very rare. Well, genetics showed it could be inherited. It's a recessive mutation. And the question is, what causes this? It appeared to be, in some versions, a single gene trait. Oh, what causes it? Well, several years ago, they found the cause of the worst one. A mutation in, I think it was in one of the lamins. I think it was lamin B. What happened when they looked at nuclei in the cells of progeria patients, and they're kind of hard to come by since it's such a rare disease, instead of the nucleus being this nice little circular thing like this, they would find that the nuclei of progeria patients were grossly distorted in shape. And that's because the nuclear lamina never assembled properly. So the nuclei had oddball shape, distorted shape. When they looked at transcription of a number of genes compared to the genes in normal people, they found that gene expression was very screwed up. Some genes were over and some genes were underexpressed. And apparently that large-scale misexpression of genes led to the symptoms of the disease. So that's an interesting thing, but that's telling you that the nuclear lamina and possibly whatever the structural roles of the nuclear lamina are affecting gene expression of numerous different genes. If your nuclear lamina isn't put together properly, you get a lot of misexpression of genes, and then you get some very nasty side effects. So that's also telling you the nuclear structure plays a role in gene expression. In this case, the nuclear lamina is a controlling factor in the expression of numerous genes. So that's the take-home lesson. So even though what we know about nuclear structure is still kind of nebulous and controversial, we do see evidence that there is something there and that it's important in regulating gene expression. Yes?
Could this have been part of the cause for the early aging of dogs while they had to put her down?
I really don't know. But the one problem is, of course, when you're cloning an organism, if you've got these epigenetic changes and you have not unmethylated a whole bunch of methylated DNA, you've not undone the epigenetic changes from a mature cell completely, God only knows what genes might be misexpressed. So that would be that could be possible, a possible contributing factor? That's a good question, by the way. But there could be so many other things that are where the expression is screwed up due to epigenetic changes that couldn't be reversed. That's probably another big factor. So it could be yes and no. Both, all of the above. Okay, all right. So we see some signs of this nuclear structure. And that somehow this plays a role in organizing the chromatin, organizing these different, you want to call them processing centers or whatever, and in regulating gene expression.